Hello, in this video we will understand about why viral infected should be in isolation, and we will learn about the evolution of viruses and infection to the human and animal as COVID-19 modification to adaptive strain. So first of all, you can understand from this picture. This is a virus and contains spike protein, nucleocapsid protein, and RNA, and spike glycoprotein, lipid bilayer, and the membrane protein, and envelope protein, and the hemagglutinin esterase, for example, for the specific enzyme. So in this video we will understand about why viral infected person should be in isolation and what is the benefit and why it is so important like COVID-19. So the first of all we will understand about the whole story and evolution and basically the virus undergoes evolution and natural selection just like cell based life and most of them evolve when two viruses infected a cell in the same person. When two viruses can infect at the same time, they may swap genetic material to make new mixed viruses with unique properties, and this unique property which that contain different type of spike protein require the different type of receptor, and the immune response still not activate because the new variant so require the new immunoglobulin against this pathogen, against this antigen. So this is the infected one person, the one strain containing virus, and the other person contain strain B, and this B contain two type of spike protein. For example, its spike protein is used for the unlocking of the our cell for infection and invasion. So remember, this is the two person, the person A and person B. It can be infect to each other through breathing, through coughing, through sexual contact, a lot of things. So these person contain different cells, and in this way the host cell, person A and B, which that contains strain A and strain B. So basically it will be infect in the cell, will replicate as well as produce the spike protein in the surface of the plasma membrane, and to bud off to produce more virus and replication to infect other tissue. So it can be basically the contamination contagious virus if by coughing, contact, and breathing, sex or kissing or some other process contaminated syringe. So in this way, it for example, this person B will MSC class one molecule, which that is a major histocompatible compatible uh, molecule, which that show that after the inflammation to show the antigen through antigen representing cell to through toll receptor, and in this way the antigen dendritic cell which they recognize and go into the lymphatic system to produce cytotoxic T cell and CD4 helper T cell, and infected cell kill through CD8, and the inflammatory mediators will produce. While on the other hand, here you can see the B memory cell. This B memory cell is basically produce the antibodies, a lot of antibodies and plasma cell. These antibodies, immunoglobulin, will against the spike protein, against this antigen, and will immune complex formation. In this way, the neutralization will occur, and the macrophage recruitment for the clearance of the debris, another infected cell, as well as neutrophil and the macrophage monocyte. So, on the other hand, that was the innate immune response, and this is the adaptive immune response. But in the when the transformation of the strain B into the strain A containing infected cell, it means the two virus, and in this way, the no antibody and with a new infection will lead to more inflammation will lead to produce different type of antibodies. So in this way the chronic inflammation will lead to cause death. So the isolation is too much important in this manner. So the antibodies generate against strain A and against but not strain B. So that is why the late strain B required. So that is why the isolation is important for the infection of the another person containing strain B or other type of virus. So let's begin to understand the mechanism. In this way, the strain A and strain B will basically the uh, genetic material will move outside and produce the spike protein will bind with the surface of the membrane and the translation of spike protein. In this way, the replication will occur. The replication will lead to RNA more production as well as the protein synthesis, and in this way the spike protein will be synthesis, and in this the bud of this vesicle and will lead to contain spike protein two type, and this two type spike protein is different as compared to the strain B because it is the rounded is the out like you can understand as well. So basically the different, and in this way the mixed of genetic material will be more adaptive for the further infection to the non-infected person or already infected with strain B or A. So in this way basically it will be bought off and the replication and more virus will be generated and the result will be the cell lysis and cell will be die or the damaged tissue. The strain B and A will combine together to form a strain C, for example, we are getting to understand. So the new infection with the non-infected person can cause and also infected with strain B and A. Because antibodies are not present already in the non-infected person, if will be cured with the strain B through antibodies naturally, but again infection can cause due to the strain C. So that is why the isolation is too much important for the evolving mechanism prevention. So this is the host infected cell is basically strain C will lead to again cytokines and inflammatory mediators will produce because it is new strain formation, the new variant like a delta variant, omicron variant, and that is why the COVID-19 is also we are concerning in this video. So the cytokines and inflammatory mediator will release again to inflammation again. So the a vaccine will not work due to the again variant. So that is why the isolation is very important for the efficacy enhancement of the vaccine. Anyhow, let's further discuss about the method of evolution. So in this way a lot of strain can be produced by the again and again infection from person to person as well as animal to animal and animal to person and person to animal. So that is why here is the strain C, D, E, F, G, H, I. By the combination and mixing of the genetic material will occur. So the isolation and quarantine is very important for the inhibition of the pandemic situation and uh, epidemic and pandemic situation. Anyhow, let's begin to understand the purpose of isolation is first is important to protect both infected persons, both infected patient. Vaccine efficacy will be increased because the variant will be decreased and basically infected and non-infected person welfare and prevention to spreading disease, prevent to evolution and prevent to mixing of genes, controlling of the further variation like COVID, Omicron, Delta variant and COVID-19 etc. That is I am mentioning the example, for example the COVID-19, but the every virus is like that. So that is why important note about that due to the seasonal virus, the influenza virus again and again attacked, um, but the already antibodies are present, but the infection will be again and again due to the modification through the birds and mammals and toward the flu toward the human, and the changes and the variation continuously will prevent the antibody from the B memory cell to non-effective more. So that is why again antibody is required to again inflammation. So thanks for watching. Bye.